here, and thanks to the organizers for the invitation. So I'll talk today about a little bit more about that external globus pallidus that's then already mentioned a little bit. In particular, I will bring a cellular perspective. I want to convince you all that one should pay attention to the tricks that neurons can play with their internal currents to be different in different conditions. So before I start, I wanted to very briefly highlight that I became a computational neuroscience actually at a very distinct point in time, which was in 1986 when I visited the second course in computational neuroscience in Woods Hole that was organized by Jim Bauer and Christoph Koch. And that was actually the first time I met Upi, because he was a TA in that course. He was a graduate student with Jim at the time and was busily programming Genesis code for reading complex morphologies into the simulator and also doing the graphical interface. And I guess he has kept that hobby ever since and is now on the second generation of Moose programming, which is, of course, a much, much enhanced version to what we had then. So the bug bit me at that course, and I decided to become a postdoc with Jim Bauer and become a modeler in my own right. And I wanted to bring to your attention that for anyone who is now young, like I was at that point, and wants to be engaged in this community, now there are actually many summer courses to choose from, one of which is still in Woods Hole, it turns out, but then another one is in Europe. It's called the Advanced Course in Computational Neuroscience in Europe. Next year will be the 18th year. It's currently, incarnation is taught in Poland. I'm one of the co-directors, and I encourage students from India to apply. So if you have questions, you can ask me at any time during the pause, and I'll explain how to apply and what the general framework is. It's a 4-week course, and you really become, you do your own research project, and you become a modeler in the course of that time. Okay, so let's start or go back to the basal ganglia, it turns out, because we already had a very nice talk on the basal ganglia. I will focus on just this one area, the external segment of the globus pallidus, and really only in rodents. And I will, so everyone draws their own diagram of the basal ganglia. In this field, it's customary to start with a network diagram of this type. And you can sort of see that I put the globus pallidus right smack in the middle. It sits a little bit like a spider in a spider web, and it has connections to multiple structures, and it gets connections from many structures. So it seems to be some sort of control center of basal ganglia activity. And much of it has to do, as was already mentioned, because the output is inhibitory, it's GABAergic inhibitory output, it has to do with suppressing the activity of other centers. And as part of the indirect pathway, as was already mentioned, probably suppresses all those movements that we do not currently want to carry out. There are a few more pathways that are interesting that we really, at this point, have very incomplete understanding. So just for a short digression, for those who are actually basal ganglia experts here and feel that everything has been said and done in this field, it's not true. There's a lot of stuff that we do not know. Just recently, so this pathway, for instance, back from the globus pallidus to the striatum was known for many years and sort of been neglected a little bit. And just recently, a new paper was published by Pete McGill in London saying that up to 25% of all GPE neurons exclusively project back to the striatum with a very, very strong inhibitory network in striatum, and even maybe doing a presynaptic trick on cortical afferents. So there's a steady stream of new insights in the basal ganglia that we still have to absorb, and there's much research still to be done. So I will not present you with a finished picture. So I will try and focus on what we have done here, which is the cellular properties of neurons in this structure. Our typical approach has been to do slice recordings, brain slice recordings from rats and mice. We can fill the neurons with a dye, and then we can, as I will show in a moment, reconstruct those neurons and start making computer simulations from them. A single globus pallidus neuron, or GPE neuron, is actually in the rodent, can span a fair amount of the total area of the globus pallidus. So there's probably some sort of integration going on, even between multiple potential motor plans, even within the dendritic arbors of one neuron, because it actually receives input from a relatively wide area of striatal outflow. It does present a challenge. So if you look at the full picture, these are not so much branched so they're very thin and pretty much unbranched dendrites that extend for very long distances. And one of the questions would be, and I will address it later on, is just how could input on the outer sides even make it to the soma? And signal transmission through very thin dendrites is actually attenuated severely. And that would be one question is, what kind of active tricks might a neuron have in order to actually be able to pay attention to stuff that happens out there? So we build a model by simplifying the soma into a sphere and basically having a 3D reconstruction with software. Usually we use Neurolucida to draw under the microscope the exact shape and put it into a computer file. Just based on first principles, because these dendrites are studded with synapses, and people like Yolande Smith have counted synapses and the density of synapses, we know that just on a very rough approximation, good enough for a modeler probably, we have on the order of maybe 1,200 excitatory synapses and about 5 times as many inhibitory synapses on this neuron. So this is also an interesting aspect in the sense that it's really dominated by inhibitory inputs, and yet if you record from living animals, it's one of the fastest firing structures in the brain. So it often goes ticking at 80 hertz, 80 spikes per second in the primate, about 30-some spikes per second in the rodent in vivo. And yet the majority of inputs are inhibitory. So there's probably some powerful ways to keep activation and spiking going. On the other hand, the excitatory inputs are actually active at a much higher frequency than the inhibitory ones. So if you just look at number of expected inputs per second, it's quite a few more actually excitatory ones than inhibitory ones. And that may explain actually a large part of that mystery. But we'll come back to the question of how inhibition excitation can control activity. Now, this is slice recording. If you just put a brain slice in a dish and you block synaptic activity and you do a whole cell recording from a GP neuron in the slice, you will find that it is a regular pacemaker. It's a little bit like a clock that goes on the order of 10 spikes per second, and it keeps going without any synaptic input. Very small steering forces, so picoamps is a small current, and if you have just 40 picoamps, you can already shut down this neuron very easily. And with just positive 40 picoamps, you can make it speed up several times. So very small control forces can create large changes in spike rate, and it is set at a steady click all by itself. Now, I'm not going to show you all the conductances, exactly how they interact to create this pattern, but let's just say that one particular conductance is important for the regularity of this firing. That is called the calcium-dependent SK conductance, which is not a voltage-gated current, but one that needs the inflow of calcium into the neuron, and then the calcium comes in with every spike, and then in response to each spike, we get a little bit of a potassium current activated by that calcium, and that spaces what we call the medium AHP, these rounded waveforms that sort of space the spikes regularly across are dependent on this current. You block this current and you get a much more irregular firing, much more bursty type of neuron. This is just, and I'm not going to talk much about this current in this talk. In fact, Charlie Wilson has written a couple of papers just on how that impacts spiking patterns of GP neurons, and you could read those. This is just to say that the individual components and membrane currents in the neuron are extremely important to control both the spontaneous spike pattern and also responses to synaptic input that this neuron may show. So we make a computer simulation of these neurons. For those of you who have not done compartmental models, I want to just use 2 slides to sort of show how they work. A compartmental model is a model where we break the neuron into distinct little pieces that are called compartments. Each of this compartment is what we call isopotential. It has one variable for voltage. And so it's basically discretization of voltage in space through these compartments. We have an electrical equivalent circuit that basically, as in terms of almost electrical engineering, describes how currents flow both across the membrane and along the cylinders, which is called the internal or axial resistance and axial current. And each type of ion channel is a parallel channel of potential ions flowing across the membrane and is governed by a set of what we call Hodgkin-Huxley equations for voltage-gated currents that basically is a set of rules that describe how voltage determines the opening and closing of these ion channels over time. We have synaptic channels that can be opened and closed by transmitters, and we have what is just called the leak, or those channels that are always open that determine the resting membrane potential. So we simulate these numerically. We still use the software Genesis, although Moose would be certainly doing just a fine job, as well as Neuron and other simulators can handle this kind of standard simulation just fine. And you do not actually have to know differential equation solvers personally that well to start being a modeler at this level. So when we put these together, we first have to quantify basically the voltage dependence of the different channels. That actually takes a lot of work to do what is called voltage clamp recordings and isolating individual channel types and their kinetics. Typically, my lab relies on other people's work for this, and we just really read the literature as carefully as we can and create basically the voltage-dependent gates for these channels and quantify them in Hodgkin-Huxley equations. And then as I will show in a moment, we have actually 9 different kind of membrane conductances in this model that are all based on experimental evidence. Then we still have to tune how much of each conductance we need. The traditional method for this is called hand tuning. So we will just up and down regulate each kind of conductance across the membrane. And we have a target, which is basically a recording, which shows us the spontaneous firing. We use the spike shape as part of our target function. And the amount of increase and decrease of firing with current injection and slices is part of our target. And we basically just twiddle with these conductances for several months until we reach a model state that at first glance looks very similar to the original recording. And then, of course, you try to publish this, and there will be many questions from the referees. And that kind of hand-tuning approach has led to many questions that are actually quite valid and important, one of which is this an arbitrary solution? How many types of models could do this? And so on. And we have, along with others, then resorted to a database strategy where we actually will so these are our 9 conductances, one of which is just calcium-gated. The other ones are voltage-gated. I will not go over the detailed kinetics for each conductance. They each have their own individual contribution to membrane dynamics and also synaptic coding. We just then randomly assign each of these conductances to one of 3 values in our study, which overall actually has 98,000 permutations of these values. And we run each and every one of those 98,000 simulations, and then we can just basically say, well, how many of the models with this complete random approach of combinations of conductance density parameters will create a useful output, and are there just a few solutions, are there many solutions, and so on. So we can basically analyze model robustness and parameter sensitivity with this what we call a brute force search approach. So the outcome of that is what, and I don't have time to, so there's a whole journal of neuroscience paper that you're certainly welcome to look up that describes many more details. But what I want to just tell you here is that, first of all, if you have biological recordings, and these are something like 130 recordings from brain slices of GP neurons, and we are just basically showing histograms of different what we call behaviors that are really just performance measures of these neurons, something like spontaneous spike rate, or the amplitude of the action potential, or the width at half maximum, or the spike frequency adaptation, which is how much the spiking gets slower and faster during a sustained input. The sag, which is a measure of when you hyperpolarize a neuron, of just how much the membrane potential relaxes back from its maximum. And a rebound ratio, which is just how much after hyperpolarization spiking is sped up compared to control. So these are just a set of benchmark measurements of neural dynamics. And first we see that biological neurons do not have a fine-tuned optimal solution to this. And I think it's important, and Sten already mentioned that variability in this spinal network is not just a flaw of biology, it's actually important for computation. And there are several folks now doing network simulations of cortical structures, and they, I think in each instance, find that variability is actually an important feature of neural computation. It's not just because we're unable to maintain tight parameters. And this variability is reproduced by our simulation space, which in reasonable, actually, shape. So if you look at these distributions, they match the distributions on the top to some degree. And we never intended them to match. We didn't optimize our parameter space in any way to optimize these matches. All we did is a random selection of combinations of these densities and plotted the 98,000 simulations that came out. So in a way, what this shows is that the biological variability could be just a random combination of conductance densities of these various conductances, and that explains a large amount of the variability that you see. And furthermore, that variability could be on the order of 2 to 4-fold is actually what Eve Marder would tell you she has found in invertebrate neurons, is that if you do quantitative assessment of currents in copies, and in the invertebrate world, they have the advantage that they can find the exact same neuron in each critter, so they can say this is the identical neuron in animal 1, 2, and 3, and you then say, well, how much sodium current, how much potassium current, how much IH does this neuron have? And it may vary on the factor 2 to 4. And if we do the same thing then in the mammal, we find that the resulting variability matches, in a first approximation, the biological variability. So this is probably the amount of sloppiness that exists. And I want to put to rest those reviewers that then say, well, your model isn't the perfect one, your model isn't optimal. We don't need an optimal model. We just need to actually match a very messy and variable biological situation. And we need to sit somewhere in that same parameter space. And we have a model that works and replicates maybe 1 out of those million biological neurons relatively exactly, and the other ones within a certain variability. So we're not trying to do perfect models. We're doing what I would call sort of the working hypothesis model that does a good job as far as we can measure it. So then we use our model for various studies of synaptic integration, and that's what I'm going to talk about in the next few minutes. The first study was done by Nathan Schultheis, who did a PhD in my lab on phase response curve analysis in the model. I will very briefly explain what a phase response curve is, and then how we found that in the globus pallidus there are some interesting features to this particular analysis and function. So a phase response curve, and for some of you this is an old hat, for some of you it might be interesting and surprising. The phase response curve is determined the following way experimentally. You disturb first of all, you have to have an oscillating background. You have to have a spontaneous pacemaking background that has a certain spacing between action potentials. That's called the phase. So the phase 360 degrees would be a certain number of milliseconds, and it would be predictable just when that next spike would occur on the own spike cycle. You make a small disturbance. In this case, it's a very brief current injection, a positive current injection, and that brief disturbance leads to a shift in the next spike timing, which is what we believe synaptic input ultimately does. It shifts spike times around. And this being an excitatory input actually left the next spike come a little earlier. Now, the interesting part is that then on the phase response curve, if you disturb the activity cycle roughly halfway, you may get a phase advance of about 5 degrees of the next spike. If you disturb the spike cycle at a slightly different place, you may actually see a much different shift in spike timing. In this case, it got much bigger. So we had a much bigger effect on the spike cycle when we stimulated here than when we stimulated here. And this is shown by the red bar. So this is a much bigger phase advance of the spike cycle. What this basically tells you is that synaptic input is dependent on what time in the spike cycle it occurs. And that actually tends to be an important aspect of things like neural synchronization and oscillations, that inputs depends and has different effects based on where in the spike cycle it occurs. We just here plot a third input to show that this function has a peak, and that if you get even closer to the spike, you get less delay again. And if you actually have an input very early in the spike cycle, like here, in this particular curve, you would have almost no effect at all. Now, there is a theoretical finding, and there are also experiments that show that some neurons have the kind of phase response curve that I just showed you. It's just got one positive peak for excitation, meaning that for all inputs during the spike cycle, we would make the next spike occur earlier. That's called a type 1 phase response curve. Then there are other neurons that have an interesting feature, which is called a type 2 phase response curve. And just if you use excitatory input, that is a depolarizing current injection or synapse, if you apply it late in the spike cycle, you get the spike to advance, occur earlier, like you expect for an excitatory input. However, if you put the same excitatory input early in the phase for about the first half of the spike cycle, it will actually delay the next spike. So paradoxically, your excitatory input will actually have an inhibitory net effect. It will delay the next spike instead of advancing it. So these biphasic phase response curves are called type 2. For those of you who want to reduce systems and understand the mathematics of this, you can actually do this with 3-parameter simulations of reduced Hodgkin-Huxley equations. You can get both types with very simple bifurcation dynamics. So it can be analyzed mathematically in much detail. We tried to figure out, well, what does the globus pallidus neuron do in terms of phase response space? We actually stimulated, or Nathan stimulated, with synaptic input various parts of the dendrite and the soma to see how much and where it would shift spikes around. And what was, I think, a new finding and generated a paper in Journal Neuroscience is that the kind of phase response curve that you see is location dependent. So for an input to the soma, this would be an AMPA-type synaptic input, an excitatory input. You would see, for excitation, you see a phase advance. For inhibition, you see a phase delay. This is a classic type 1 phase response curve. However, if you put the same synapses, the same AMPA synapses on a distal dendrite, you actually find that it converts into a type 2 phase response curve. So phase response curves then are location specific, and that was a new result. And that would mean that excitation to a distal part of the dendrite could have a completely different effect on things like network synchronization between pallidus neurons than input on the proximal part. And if one tries to do reduced network simulations, one would want to keep those factors in mind and adjust those simulations to reflect these aspects. Now, how did this? So the model, obviously, you should be able to understand why this is happening. And this is one of the good ways to use models, is to delve into the mechanistic underpinnings, the conductances that cause these effects. This was is a relatively easy effect to understand in this model. So this is our phase response curve for a synaptic excitation in the distal dendrite. And then if you block or up or down regulate the SK current in the dendrite, you can create various amounts of the type 2 character. And if you entirely block the SK current, and this is just local to the stimulus, so if you just take away the SK current from this part of the dendrite, you actually convert this into a type 1 phase response curve. So in this case, basically the SK conductance, and it also requires the high voltage gated calcium current as an inflow of calcium to trigger the SK conductance. So it's the local depolarization of the stimulus that results in a calcium inflow and then an activation of the potassium current that ultimately delays the spike. And it turns out that the steering current, the current that I put in as a synapse or as a current injection, is much smaller than the secondary current that I trigger through the calcium inflow. And this is how I can have a net inhibitory effect from an excitatory input in this neuron. So that's the mechanistics of that, and we wrote a second paper about how this may all impact in vivo patterns and complex synaptic inputs that I don't have time to talk about. So I want to go on to the next story, which was actually done over several years in the lab and started with work by a graduate student, Jesse Hansen, who worked together with myself and Yolan Smith at Emory University, and we were interested in just how distal dendrites could potentially even make their effect known at the soma. We thought we need some sort of amplification of signals on the way. One natural way to amplify signals is with inward currents in the dendrites, currents that can prolong and increase depolarization In an extreme case, you can actually create dendritic action potentials with sodium carns. That was already known to be the case in mitral cells that, for instance, UPI works on. In the olfactory bulb, that's known for quite a while. And then in pyramidal neurons, it is also actually under debate whether forward propagating action potentials occur, but certainly backward propagating action potentials occur in dendrites. So for those of you who sort of just have a classic introductory neurobiology, the soma is supposed to have action potentials and the dendrite is supposed to listen to inputs. Well, in real dendrites, some neurons actually have action potentials in the dendrites. And we wanted to A, know whether this may happen in GP, and B, if it does happen, what consequences would it have on computation. And we, again, resort to a mix of physiology and computational approaches. So the physiology is shown here in just one slide from this paper, that if we record from the soma and we basically put a fluorescent dye in the soma so that we can see the dendrites after 10 minutes in the slice, and we can just snuggle up to a dendrite here, very, very close, just a few microns away, and we can either puff glutamate here or we can electrically stimulate the local inputs. We can, and we put this actually in voltage clamps, so we don't actually let the neuron depolarize to threshold. We keep it away from threshold, hyperpolarized. Then we see actually that our synaptic stimulation here leads to an arriving action current at the soma at a certain threshold. And if we hyperpolarize even more to a very negative potential, minus 100, we basically see no synaptic current reach at all. So this is positive evidence for the induction of dendritic sodium action potentials that then run into the voltage clamp at the level of the soma and generate an action current there. So this doesn't happen in all dendrites or all neurons, so there seems to be a mix of GP neurons that can or cannot have strong enough local currents to trigger action potentials. And Jesse Hansen also worked with sodium channel antibodies in Yolan Smith's lab, which is an EM lab, and he found evidence for antibody binding of 3 distinct subunits of voltage-gated sodium channels in the dendrites of GP neurons. So then Jeremy Edgerton, who's a postdoc, or was a postdoc in the lab, did a modeling study. So he used the same GP model. By the way, that model is available on NeuronDB at Yale. So if anyone wants to download and play with this model, it's publicly available. You would want to run Genesis unless you want to translate it to Neuron yourself, and you can start playing with this neuron tomorrow if you want to. So what Jeremy did is he created a family of models that had a different amount of dendritic sodium channels. So the uniform model had the most dendritic sodium channels. In fact, they were uniformly distributed throughout the soma and the dendrites. And then he created various gradient models where the steepest gradient basically had almost no dendritic sodium channels and had a very rapid transition from the soma into the proximal dendrite to no sodium channel density in the distal dendrites. He verified that back-propagating action potentials were fully present in the uniform case, and that we basically had just passive, highly attenuating propagation of action potentials in the models that had a steep gradient of sodium current. Now, are these then completely differently behaving models? And would both of these models be compatible with the kind of recordings we'd obtained in slices? And the answer is yes. So if you simulate a neuron with low or high sodium current in the dendrites, you actually can tune them very easily to both have the same spontaneous spiking frequency and both have a very similar Fi curve, meaning if you just inject current in the soma, they speed up or slow down the same amount. So from the point of view of a somatic recording, they behave identically almost, except that you see that these guys have a more rounded after hyperpolarization, and these guys have a more sawtooth kind of after hyperpolarization with the highest sodium concentrations. And also the one with the lowest sodium concentrations tend to end in depolarization block more easily when you try to depolarize them strongly. And if you look at our slice recordings, and this is just parallel universe. We can't prove it's due to dendritic sodium currents because we have no independent measure of those in our slice recordings. But our slice recordings show some neurons have this rounded shape to the AHPs. Some other neurons have this sawtooth shape to their AHPs, and we also have a large amount of variability of how much current they can take before they end up in a depolarization block. So there seems to be a parallel universe in our slice recordings to the potential amount of dendritic sodium current. So then we wanted to know, well, what is the consequence for dendritic sodium currents on synaptic integration and potential network function? And we then basically put in vivo-like input patterns onto these neurons in the model and sort of wanted to see, well, what is controlling spiking and what does the outcoming spike pattern look like in these different models. Now, if we have almost only sodium current in the soma and none in the dendrites, then all spiking is actually triggered in the axon hillock. And it turns out that you get a fast and regular firing neuron even if the inputs are equally, they're Passuan distributed random inputs, and we have 485, I believe, synapses along the entire neuron. And we get this fast-firing regular. The autocorrelation here shows side peaks, which means that each spike occurs at a regular interval to the next one, which gives you these, what we call a side peak in an autocorrelation, tells you it's a regular firing neuron. If you have a uniform dendritic sodium current, you actually get equally fast firing. In fact, we will show a faster firing, but you get a much more irregular spiking, and you don't get the side peaks in the autocorrelation. So you're going to deregularize the firing. Now, what about synapses that actually cause the spikes in the different models? And this is an interesting shift. So for the neuron that has only somatic and axonal sodium channels, maybe to no surprise, and in green are now synapses that are effective in causing spikes, and in blue are synapses that are not effective in causing spikes. What you see is that the only effective synapses are those close to the soma, because they will create big EPSPs in the soma, and therefore they can induce spikes. So this is what we would call a general summation device. So all the currents are summated and increase spike firing frequency in the soma. The inputs close to the soma are the ones that work. Now, the dendritic spiking model actually is the total opposite. The synapses that are effective are the ones on the most distal ends on the dendrites, because they are actually sitting at what we call a high input resistance side of the neuron. They will trigger the biggest EPSPs in the whole neuron, and the biggest EPSPs will cross threshold for action potentials. And then these guys are privileged in triggering local action potentials in dendrites. So now you have the opposite, and you would actually have to have some homeostatic plasticity mechanism to try and regulate what we call a democratic model that would have just the right amount of sodium current to give about equal weight to each synapse. So, and I just very briefly want to show, there's a little video of basically, this is the firing of the model with all axonals. So the inputs are measured. This is just a local measurement of voltage in the different dendrites. This is the somatic channel where the action potentials are initiated. And you see a very regular firing pattern and basically just a twinkling of depolarization throughout the dendrite that gets integrated here and sums up to the action potential. Now we have the dendritic spiking model, and you see that it looks completely different in the sense that you basically have hyperpolarization, and then there's a local set of inputs that exceed threshold. It'll be a firing, and then the firing first forward propagates and then backward propagates through all the other dendrites, and you have these very distinct locally triggered events. And you see now spikes throughout the neuron. Now this type of neuron then is really a coincidence detector. It spikes in reaction to local coincidences in specific dendrites, which is a very different computational function from summation. How that plays a role in, we have not done network simulations, so I cannot tell you how that would actually change things like action selection, but it's certainly worth thinking about. We did a brief study on just how this may involve Parkinsonian activity patterns. So in Parkinson's disease, the globus pallidus changes activity patterns from basically highly uncorrelated firing. Usually it's irregular, and some cells are more regular. But if you look at two neurons recorded at any distance from each other, their spike timing is not related to each other. That's called decorrelation. In Parkinson's disease, then actually globus pallidus neurons and other parts of the basal ganglia start to be what we call hypersynchronized. And we see these cross correlations where the spikes of the different neurons are highly correlated in time. And this is actually some sort of pathological synchronization that may be underlying a fair amount of the disease problems. And treatments such as deep brain stimulation may actually break up the synchronization more than doing anything else. And that seems to be beneficial. So we were just interested in how those sodium channels may or may not contribute to synchronization or the lack thereof. And we got data from Pete McGill in Oxford, who gave us his subthalamic nucleus activity patterns from 6-hydroxydopamine lesioned rats that show this rhythmic bursting pattern. And we post-processed those spike trains to create 800 inputs to our globus pallidus neuron model that were then beta band modulated synchronized input trains. Since I'm running low on time, I'm going a little faster, but it's a beta modulated synchronized input pattern to our globus pallidus neuron. And then we put that Parkinsonian input pattern to either the sodium dendritic sodium spiking or the non-dendritic sodium spiking model. And we found actually that the dendritic sodium spiking model, and this was a surprise, it was the opposite from what I expected, it turns out. And this is where modeling actually sometimes guides your intuition and does not do what you expect it would do. But the one with the high dendritic sodium cons actually led to less permeation of oscillations, and the one with low dendritic and all axonal spiking actually amplified or led oscillations through more easily. So it is conceivable that a shift in sodium channel densities with Parkinson's disease, and a lot of these conductances are dependent on modulatory state and homeostatic regulation. So it's very feasible that these parameters shift in Parkinson's disease. So a prediction could be that a shift towards less dendritic sodium spiking would lead you to more oscillation synchronization in the basal ganglia. In the last 2 minutes, just as a sort of that we're moving on. So in fact, we are currently no longer working on globus pallidus neurons very much in the lab. We have moved on to the thalamus together with some other folks at Emory. We have a Udall's Parkinson's Disease Center grant. And we're now looking at the thalamus, mostly the motor thalamus. So we're moving on to the output of the basal ganglia to the motor thalamus, VM in the mouse, and trying to figure out how basal ganglia outflow directs spiking patterns in the thalamus, which then would ultimately affect spiking patterns in the cerebral cortex. Just as a little introductory story, another interesting model organism for basal ganglia function has been the songbird and the learning circuit, the song learning circuit in songbirds. And David Perkel, who was actually a fellow postdoc at Caltech when Upi and I were there, so we know him well from then. But he is studying bird brains and found out that the basal ganglia analog of the bird brain area X can cause, in the bird brain analog of the thalamus, DLM, can cause what we call rebound spike bursts. That is, a strong inhibition can actually hyperpolarize the cell so as to de-inactivate some cons that then cause a burst pattern. And this is actually known from the mammalian thalamus for many years, that we have these what we call T-type bursting performances. If you hyperpolarize a thalamic neuron and you let go of the hyperpolarization, you'll have this T-type calcium channel dependent rapid burst. This is a recording that we did in slices from VM thalamus in mice. Then we used optogenetics to add inhibition from basal ganglia outputs. So we transfected the substantia nigra reticulata in mice with stenorodopsin, and then we can record in the thalamus and just optically stimulate just the inhibitor inputs from the substantia nigra. We get nice IPSPs, and we can then ask the question of those are suitable or not suitable to create rebound bursts. The maybe surprising answer is, and again, it's maybe not what would be the nicest story to get, but the answer is that mostly it does not generate rebound bursts. Mostly we get classical inhibition, so there's no spike bursts after these IPSPs. And then when we drive the cell to spontaneous spiking, the IPSPs do what they're supposed to do, is basically lead to short interruptions of spike timing. So we can actually find only in very narrow parameter regimes, and so if we tweak it just right and give little bursts of optical pulses and adjust at the right membrane potential, then once in a while we can actually get rebound bursts. But again, in the upstates, they just turn into pauses. But the parameter regime for rebounds is extremely narrow, and in our hands would not really be the one we expect to operate in vivo very reliably. So we're back to the more classic inhibitory model of basal ganglia function. So as conclusions, globus pallidus single-cell models can replicate brain slice data variability through random conductance densities. That was the brute force database studies of Cengiz Günay and Jeremy. The synaptic input-output function is likely location-dependent, so where you stimulate the dendrite is important for what it does to network properties. And specifically excitatory input to the distal dendrite gives you type 2 phase response curves. Physiological recordings indicate that the GP neurons are capable of dendritic sodium spike initiation, and in a model, such spiking has important consequences for synaptic integration. And we basically switch the neuron from being a summator to a coincidence detector with dendritic sodium spikes. Parkinsonian synchronization is reduced in the presence of dendritic sodium spike initiation, and integration of basal ganglia input in motor thalamus most likely reflects the traditional model of hyperpolarizing inhibition rather than rebound spiking. And for the very last slide, I want to show the folks who did the work. Jeremy Edgerton's been a postdoc for several years. Jesse Hansen did the original work on sodium channels. Eric Hendrickson's work I didn't get to talk about. Cengiz Günay did the database, and he's here with Nathan Schultheis, who did the phase response curves. And that's just one of the parties in my house that happen when there are birthdays and other things. So thanks a lot for your attention.
So question is about GPE. It receives massive inhibition from striatum. And yes, I agree that SDN inputs are excitatory, so it makes sense to talk about what will happen to the PRCs. But in connection to these excitatory inhibitory inputs, how do you think these PRCs are going to tell us something about oscillations? Or how they change actually when you get excitatory inhibitory current simultaneously?
Okay, so this is we did do a follow-up on the study I showed where we did put a background, and in fact it is a good question, how do phase response curves even matter when there isn't an oscillatory background but a random stochastic background steered by many inputs? And it turned out that the average behavior of the response was still the same. So if you did a couple of hundred stimulations, you would still, on average, see the same shift in spike times that was phase-dependent of the input. It's in an average sense, it still works. In individual stimulation, it depends very much on what the random inputs are at that point in time. Now, inhibition we did not do phase response curves for inhibition per se, but the inhibition would just create the noisy background that would basically take away from the reliability of these phase responses. There's room for many more studies.
Just did one short one. PRC is actually a property of the spiking mechanism. And now these distal inputs are affecting the PRC. Does it mean that the model we should think of, like, we should discard the point model? Or I mean, why is it actually happening that distal inputs are changing PRCs? Is it the input that is coming has changed?
Well, so PRC, so one has to delve deeply into the theory of PRCs and whether it is or isn't valid for biological neurons. Usually we think about weak inputs, inputs that do not depolarize the neuron enough to actually activate voltage-gated conductances, and only then is it the spike initiation mechanism that determines the PRC. If you start depolarizing or hyperpolarizing the neuron enough to activate and inactivate other conductances in the membrane, then they also play an important role. And it turns out that, I mean, I'm sort of the realist that tries to see what really happens in biological neurons, and most inputs are big enough to significantly interact with all kinds of voltage-dependent mechanisms in the membrane. And it isn't just the pure PRCs that are the weak input. And then the theory has a hard time being fully predictive of what would happen in networks. That is also true. So we cannot now hand this over to a theorist to say what happens with networks. You probably have to simulate it.
Okay, very nice talk. 2 questions. First, do you know the distribution of KCA channels along the dendrites? I mean, some neurons have rather little, most of the KCAs on proximal dendrites and little on distal dendrites.
Yeah, so we do not know the accurate distribution. So we don't have antibodies, so we have certainly not used them if they exist. So we're not sure. It has been a little bit has been done on calcium corns, and you also need the calcium inflow, obviously, to trigger SK. So and the calcium corn is appropriately located in the dendrites.
And secondly, you mentioned in passing Pete McGill's new findings
right.
of 2 types of neurons, and they are, one is feeding back very much to stratum, but the input is rather is not well known, except that they are alternating with the sort of GPE
right.
STN projections. Do you know if, and they have different morphology also, do you know if your cells are primarily the STN projecting, or?
No, we do not know that. So, and the morphology is different if you already know which one they're belonging to, but they're widely overlapping. So you cannot,
okay.
by just looking at the shape, say they're this or the other type. So they're only sort of statistically significant small difference in morphology. It's not like they're really looking different at first sight.
Okay.
So no, we don't know is the answer. So, and it could be that one of them is more sodium spike prone than the other. It's entirely possible. But we would not know at this point.
So you showed that small perturbations, 40 picoamps or so, can change both the rate and the phase of the output, and it's also location dependent. And so I was just wondering, this level of input just represents a few synapses out of the thousands that are there, or could represent just a few synapses.
Yeah, I think that.
Given that synapses can be plastic, I guess my question is, what maintains a set point? I mean, you could easily imagine that this whole system becomes extremely wandering out in nonlinear space. Is there kind of a global minimum, and how do you maintain that?
Well, these are all good questions, and the answer is usually that we don't know enough yet about the system. So we believe, and I think the whole field is studying, not just in the basal ganglia, but in the whole brain, is studying homeostatic plasticity mechanisms quite a bit. And Gina Turnigiano gave a nice talk at SFN, just as the tip of the iceberg, that just about every synaptic input strength and every conductance we know of undergoes some sort of activity-dependent regulation to keep it within its parameter range, to not make the neuron be excited too much or too little. So we obviously, I think that happens in the globus pallidus as well, but we have not studied that, and it's actually, that would be a lot of studies to try and figure out exactly how many homeostatic regulatory mechanisms there are and what they do. And the other hidden, and you're absolutely right that in terms of really understanding basal ganglia function, we would want to know the status of all synapses and how many of them are strong and how many of them are weak, and what learning does to the population of inputs, and how many of them are now contributing, how many of them are not contributing so much. So I totally agree that the long-term synaptic plasticity is where these networks get trained, and where certain ensembles can be established as important and representing a certain behavior. And while we know that there's long-term potentiation and depression in all of these circuits, we really don't know how they're related to motor learning at this point. So I think we're sort of slowly coming from the input side. In the stratum, the ideas are a little more clear, and the plasticity mechanisms have been studied a little better, and we have some ideas and models how that impacts learning. And when it comes to plasticity mechanisms in the globus pallidus and how they contribute to learning, I think everybody's ideas are quite vague at this point. But I think the answer is that they will be important.
Okay, so we'll have one more question and then have a tea break now, and it'll be a short tea break to try and catch up. So one more question, and then maybe we can reconvene at 11:25. Okay, so there was one more question, and then tea break.
Thank you, sir, for the talk. Actually, you were mentioning that the beta oscillations come when the sodium densities and all go down, but also when there are more lateral connections in STN, you get beta oscillations. So which is actually initiating it? Is it from the GPE or?
I get all those questions that I have no answers for. So the triggering of beta oscillations in Parkinson's disease is under intense study, and there are many people who have favorite ideas. So one favorite idea is that there's a core oscillatory mechanism between the globus pallidus and STN, and it requires really the feedback network. You couldn't say it's one or the other. It's the feedback connections that cause the beta in that model. And it was popularized originally by Dietmar Plenz, even before we knew about it happening in Parkinson's disease, because in co-cultures of STN and GP neurons, you can get these ping-pong effect of oscillations. But then there are folks like very serious mathematicians like Nancy Coppell who would blandly put it in the stratum as the cause, as the ultimate beta oscillatory mechanism. She has very specific GABA synapse time constants in mind for causing beta oscillations. So there's at least a straddle and a GP-STN view of what causes them. I once saw a very interesting talk from a physicist who just tried to decompose the multiple sources of oscillations in brain networks with multi-unit recordings in awake animals. And he came through some mathematical analysis, I don't remember, to the possibility of several hundred oscillators in the brain. So there are many mechanisms that can cause oscillations. And I think there's another likely source in cortex itself for beta as well. And it's been shown that subthalamic nucleus is influenced strongly by cortex in its activity, and maybe some of the oscillatory patterns actually come from cortex. And it's not mutually exclusive. You can actually have multiple pattern generators, and they can become synchronized and reinforcing, or they can not become that way in certain conditions. So I don't even think it's necessary that there has to be one source of beta oscillations. But I don't have a personal favorite. I have high estimation for all the folks who do these different studies.
Yeah, so tea and coffee has been arranged right outside the auditorium. So while you're having a tea or coffee, please pick up your name tag if you haven't already done so. So the name tags are now ready. And those of you who are being paid travel support, you must have already got the form. Please fill up your form and give them back to me so that I can give it to our office for processing. All right, so we'll get back at...
Okay, let's get started. It's a pleasure to introduce Dr. Bapiraju from